So hello, everyone. Hello. So thank you guys for coming to our Arima workshop. So my name is Kevin. I am the Strategic Account Executive for Arima Genomics for the Northeast. I know a few of you already, but very nice to meet all of you, and thanks for coming. Today is going to be a high-level technical overview, of the technology. And we have two great speakers as well, and I'll introduce them at the end of my talk. And so just a brief outline on our of this presentation. So just an overview of the technology, introduce some of the products and services that we offer, the benefits of our technology for 3D genomics, and then kind of overviews of some of our specific products, our Genome-wide Hi-C, FFPE, Hi-C for FFPE, our Capture Hi-C promoter, and oncology panel as well. And then also some information on how to get started. So just a brief overview. So Hi-C can reveal spatial and temporal changes in chromatin structure that have been shown to alter gene regulation and cellular function. And our off-the-shelf Hi-C technology allows you to use Illumina next-generation sequencing to gain unparalleled access to the 3D genomic landscape of any genome. And so we can see that. So with 3D genomics, we're able to utilize sequence to identify structural variants, as well as detect 3D interactions across your genome of interest. And so we offer kits as well as services. And so we have a wide product portfolio of kits. So we have a Genome-wide Hi-C, Hi-Chip, promoter capture, custom capture, as well as genome assembly. And so I'm only going to touch on a few of these today, just for the sake of time. And also we offer end-to-end service. So from start, so we can send us your sample and we can work it all the way down to bioinformatic analysis and send you that data for you to look at and publish. Why choose Arima Genomics? So we have a robust Hi-C technology, integrated library prep, end-to-end solutions, user-friendly workflow, as well as flexible input amounts and world-class support and service. So you won't be alone throughout the entire workflow. We'll be able to help you every step of the way. So why choose Arima over a homebrew purchase? So it's more cost-effective to scale. So our kits are priced for running multiple experiments. So it's more efficient and cost-effective than homebrew methods, ease of use. So it's developed and optimized, so you don't have to troubleshoot. And you have support as well. So you'll have the support of a technical support team as well as our bioinformatics team. So to talk about Genome-wide Hi-C briefly. So our Genome-wide Hi-C allows you to understand gene regulation and epigenetic mechanisms and discover how spatial relationships in your genome impact the cellular development and disease processes. And so you can identify structural elements in the genome, as well as compartments such as tags, and as well as loops. And so here's an example of the power of Hi-C 3D genomics from the Giorgio Galli group at Novartis. So they show that estrogen receptor exerts its oncogenic role in breast cancer by engaging this transcriptional regulatory element that's enriched in TFAP2C. And the figure here, which is kind of a little hard to see, but it highlights how Hi-C 3D genomic mapping revealed how TFAP2C is a critical target for the estrogen receptor through its interaction with TFAP2C's transcriptional enhancer TET2. Sorry, not TET2. So here is a overview of what the workflow looks like for our High-C technology. So we have flexible input amounts, so you can use various amounts of tissue, blood, cells, etc. as input. The workflow is fairly simple. So once you cross-link and extracted your DNA, you would digest with a restriction enzyme cocktail, label the ends with biotin and ligate them, purify your DNA, and then we proceed to the library preparation. So you would shear your sample using a diagenetic Koparis device, ligate your sequencing adapters to these sheared samples, amplify, and then run on your Illumina sequencer for deep sequencing. There are also shallow sequencing QC steps as well that are predicative and can give you a good estimate of the quality of your library before you do any deep sequencing as well. And we also have open source bioinformatic pipelines. So these are open source tools that use FASTQ files, and essentially they spit out visualization and reports of your data. So next are FFPE. So we have an FFPE for High-C. So you can use your fixed samples to detect non-coding and coding structural variants and identify novel gene fusions. You can study fusions that have been archived in FFPE specimens from greater than 10 years or so. Here, just kind of a comparison of our FFPE High-C compared to fresh frozen sample. And just to kind of briefly go over this, but essentially this shows that there's a high correlation between match frozen samples and our FFPE samples. So you're not going to get diminished results because you're dealing with FFPE. You're going to still get the same high quality High-C library. Here is an example of how our High-C pipeline has been used, or how our High-C workflow has been used to identify fusions and complex rearrangements. So in A, we have a genome-wide High-C contact map of a breast tumor, contact matrix on a breast tumor. And when you zoom in B, you see that there are 3D interactions between chromosome 15 and 12. And this, the punctate signal around the lower left of the matrix is indicative of complex rearrangements between cells. And so when you zoom in a bit more, you can see that there is a fusion between ETB6 and NTRK3. And D is kind of a schematic of this rearrangement and these fusions that were detected using our High-C technology. And so our High-C, so we were able, so this figure we're able to show that using orthogonal assays, we were able to identify the same gene fusions, as well as identify fusion partners that weren't identified or recapitulated by these orthogonal assays, discovered by orthogonal assays. So our assay seems to be a bit more sensitive than some of these typical approaches for looking at fusions. And so the workflow for FFPE is pretty much the same as for High-C, except that upstream from your block or your scroll, there's a dewaxing step. And that's pretty much it. And then you go straight into digest with the restriction enzymes and so on and so forth through the rest of the workflow. We have an SP pipeline, which is also open source. And yeah, just to let you know. So you can also use this as well to look at these breakpoints in your High-C data. And depending on the quality of your FFPE High-C data, you could also do some look at compartments, tags maybe as well. So you're not just limited to detecting SPs. So next, I'm going to go over our capture High-C, so our targeted approaches. So we have a couple of types of capture Hi-C. We have promoter capture that pretty much has probes that are tiled across promoter interest. Variant capture, so you have these probes against your variants of interest. And then the region capture, so if you have a construct or something or an insertion somewhere and you wanted to see the interactions between that construct and the rest of your genome, you could do something like that, region capture, or if you're looking at a specific region of interest. The interactions between one region to a specific region of interest. And so to illustrate, so why use capture-based approach? So you can understand long-range gene regulatory mechanisms, test hypotheses formulated by genome-wide DNA analysis, and increase the resolution across the regions of interest, as well as reduce costs and increase computational efficiency. So to illustrate that, we're able to illustrate these benefits of capture. So in the top section, we have a genome-wide Hi-C, genome-wide Hi-C interactions around this gene PRR5L. And you can see that there are a ton of contacts in this 10 kb region. And but when you kind of take away some of these other contacts that are surrounding this region and only focus on the ones that are connecting to the promoter, you see that there are a lot less contacts. But when you can gain a bit more insight and enrich for and get higher sequence coverage using promoter capture that just targets your essentially that's baited on your promoter, your PRR5L promoter, and you can see all these other connections strongly and without any kind of backgrounds as well. So just to talk about promoter capture, so we can characterize regulatory differences between samples, generate profiles across promoter regions, and it's fairly flexible and robust. We have our off-the-shelf promoter capture kits for human and mice that are fairly comprehensive with over 23,000 gene targets. The workflow is the same, except so you prep your sample, do your Hi-C library. The only difference is that there's an enrichment step with your probes during the library preparation or after library preparation. We also have open source tools as well for analyzing your capture data as well, or doing promoter capture. So we recently launched our oncology panel. And so we have this panel that targets about 1,400 genes that have been shown to be drivers of cancer. So here is a bit of a breakdown of the genes that are being targeted. So we have some heme. So we have a good portion of these panels made up of genes from heme genes, so hematological genes that are connected to hematological conditions, as well as actionable cancer targets, tumor suppressor genes, fusions, etc. So these are all collected from several oncogene databases, so it's fairly comprehensive, and we're launching it sometime in October. There it is. Now let's get too far. All right, almost done. Yeah, it's just that one particular stuff. Yeah. Anyway, so just to kind of here we're able to detect the fusion of. Sorry about that. All right, perfect. This slide. So here we're able to identify a known fusion. So for BRCA and ABL, using our Hi-C technology. And so with our capture panel, with our oncology panel, we're able to capture we're able to capture this this fusion, this known fusion in our genome-wide Hi-C But that requires about over 175 million reads. With our oncology panel, with only 4 million reads, we're able to still get that same high resolution information and detect that, that fusion. So here is just a comparison of our capture panel versus other types of our oncology panel versus other types of DNA and RNA panels. And just kind of a brief overview. So you can detect 50 kb. So breakpoint resolution is about 50 kb. And we're able to detect known fusions as well as novel fusions for discovering regulatory elements. And we're still improving and developing the kit to offer more and provide greater resolution as well. So how to get started? So you can reach out to me or come to the website, request a consultation. We'll set up a call. We'll talk about your project and see what kits or services fit best for your research goals and your budget. We also have, once you purchase, we'll have access to our technical support team. We'll have a kickoff call with them if you want, and they'll go over your workflow with you and discuss any kind of technical hiccups you might, or technical concerns you may have about using our technology. All of our technical documents and everything are on our website. So documents are publications as well. And that is it. Any questions? Yep. Yeah.
Can you comment a little bit on what the input is for the promoter capture Hi-C?
Yeah, so the promoter capture Hi-C is fairly flexible. It only requires, so like our genome-wide, regular genome-wide Hi-C, all you need is about 1 million cells at least, or about 100 to 150 like milligrams of tissue.
So 1 million cells.
Yeah, 1 million cells. Yep.
So far you have the promoter panel and the oncology panel. Do you have something where you can set up your own regions of interest?
Yes, so I didn't mention that, but we also offer custom panels as well. And depending on the size of your panels, you know, we have panels as low as that cover about 1 kb to half a megabase to panels as large as 24 megabases in size. So we can, you know, have a discussion. You just send us your design, or you send us your regions of interest, and we would design the probes for you. And then with the kit, you would just get everything, our Hi-C reagents, the probes, and the NGS library as well to do that. Great question. It's proprietary, but I can send you information about the cut sites, though. We can let you know about that if you wanted to do like in silico kind of things. So look at where those breakpoints would be, or where those cut sites would be. Yeah. Anything else? If not, we can get to the real reason why you guys are here, is to see these great talks. So our first presenter is Ceyda Durmaz, who is at the Weill Cornell Medical College. And after Ceyda is James Satterlee from Cold Spring Harbor. Thank you, guys.
Okay, great. So today I'm going to be talking about Histone H1 deficiency and how that can disrupt 3D chromatin architecture to promote a more aggressive B-cell lymphoma. So, great. So in chromatin, linker histone H1 is a protein that binds linker DNA. So let me see. Great. It binds linker DNA, and it's critical for assisting chromatin to fold into higher order structures. Essentially, H1 acts as a transcriptional repressor, and it limits chromatin accessibility and is depleted from actively transcribed regions. So mice and human both express 10 H1 isoforms, and there are 5 isoforms in particular, H1A, B, C, D, and E, that are expressed in a replication dependent manner. So while H1 is commonly mutated in many types of cancers, B-cell lymphomas in particular have the highest frequency. And B-cell lymphomas mutations in H1 are specifically high for B through B, which is here. So, right, and this is also impressive given that B-cell lymphomas are highly heterogeneous and have a high somatic mutation burden. So in one of our recent studies, which is about 2 years ago, we looked at the mutations for the H1 isoforms B through E, and we here using whole genome sequencing, we found, sorry, there's the mouse. These are driver mutations, and the histone H1 isoforms C and E were among the top that were driving the mutations in the lymphomas. So one of the questions is how these mutations would drive lymphoma programs more mechanistically. So basically my talk today is just going to cover these 3 main points. Is how does H1C and E deficiency impact transcriptional programs to promote that more aggressive phenotype? And can the reorganization of compartments play a role in reorganizing that transcriptional reprogramming? And do compartment shifts rewire promoter interactions to promote that gene expression? So the second question will be answered using HiC, and the third question is inferred through promoter capture HiC. So here's just an experimental setup for how we do our experiments. So we used FABP BCL2 mice model, which models DLBCL or lymphomas subtypes, and it has a high expression among BCL2. And basically, we took the mice and we used SRB's sheep red blood cells to start forming these germinal centers. And essentially, when you look at the survival curves, focusing on these 3 particular. So these are the wild types in just a normal setting for the mutations. And here, blue is the wild type with lymphomas. And you can see that there's a decrease in survival for those with the depletion of H1. So looking at the transcriptional programs here, this is just RNA-seq data, and these are just showing the trajectories of the differentially expressed genes. And essentially from these clusters, you have several different trajectories. Most of the genes are increasing when we found with the H1 depletion. And looking at the geoterms for the genes in each of these clusters, there are different programs that are being upregulated versus downregulated. So cluster 1 seems to be more upregulating some plasticity. It's filled with more neuronal and stem cell genes, and similar things happening in cluster 2 with more stem cell genes, but also with involvement in movement of the cells, which corresponds to our phenotype that there's also been the invasion to other types of tissue with this cancer in the mice. And cluster 3 is more of a strong expression for these immune system responses. And cluster 4 is interesting, particularly because it seems to be promoting more of that cancer phenotype to enhance the fitness of the cells, where DNA repair, double-strand break repair, and cell cycle checkpoint genes are downregulated. And in addition, there's also a downregulation of chromosome organization genes, such as those that are organizing the chromosome during cell cycle and also condensing the chromosome. So using, this is the other model. So that was for the RNA-seq. This experimental design is specifically for the HiC and promoter capture, which are here from the same mice. So here we also took the mice, irradiated them, and immunized them with the sheep red blood cells to get those germinal centers and sack them at several different days. And this was just genotyping to show that we do have the correct genotypes for the mice. So H1 wild type in the lymphomas, H1 double knockout het is for the heterozygous for H1C and H1E, and double knockout homo is homogeneous for H1C and H1E. And then just looking at some of the phenotypes, so these are just gated on B cells, and then plasmablast and plasma cells, which are the next cells that come after B cells differentiate post-germinal center. So there's a slight decrease in the B cells, and then there's also an increase in the plasma cells, which is suggesting that the lymphomas are becoming more aggressive. Let's see. So here, this is just Hi-C data, and this is showing compartment shifts. So we identified several different categories of compartment shifts. So if you look here, you have like B to A. B is more condensed, A is more open. And you have a couple of different shifts where B is becoming from strong B and becoming more open. You can have a complete shift, switch directly from B to A or A to B, and then several other gradients switches. And here, these are just different differential compartment switches looking at the C-scores from Hi-C. And you can see that from wild type to het, just the heterozygous dosage drop doesn't induce as much switches, but shifts. Whereas once you go from het to homo, there's this large increase in those that are already condensed. So the part of the chromosome that's closed is starting to become more open with the depletion of H1. And what's also interesting is that there's also some that are in the open part of the chromosome regions that are becoming weaker. So that could possibly like indicate that there might be also a shift of where H1 is going. But let me see. And so here, this is also incorporating the Hi-C with the RNA-seq. And these are, again, the different shifts. So with the B to B wild type, I'm just going to point out a few like key differences here. Also, I don't have the statistics here, but everything was compared to the stables, and they were all significant for differences. So with those that are, these are ones that are blues, those that are compacting, and then red is showing what is decompacting. And on the y-axis, we have the fold change between whichever condition genotypes here. So for those that are decompacting, we have a large number of genes that are already in the B to B area that are becoming weaker B that have an increase in gene expression, which was interesting, which might potentially, we haven't looked into it further yet, but might have an impact on how the het and homo interact, because for het and homo, we get what's expected. For those that are decompacting or becoming open, show an increase in gene expression. So this is, I'm going to switch over to the promoter capture now to answer that other question, is whether these genes that are involved in those compartments that are switching are also gaining interactions from the promoters of those genes to other regulatory elements such as enhancers. So here, this is just the layout of the workflow for promoter capture, but Kevin already went over it, so I'm not going to do it again. So here, this is just a brief summary. Basically, after doing the capture, you get about 120 to 150 million significant loops. There's more, but those are the ones that are deemed significant and not noise. So, and of those, you can also, you have a statistic which you can look at the number, the type of interactions. So there can be two. There's going to be promoters interacting with promoters, or promoters interacting with regulatory elements, which is going to be known as other ends from here on. So here in the purple is the promoter to promoter interaction, and here in the orange is the promoter to other regions interactions. And from wild type to het, you've got about a 50% like equal ratio for those. But once you get to the homo, there's an increase, there's a slight increase in the interactions of those promoters interacting with other regulatory elements. So this also opens up the thought of what my lab in particular was interested in using the promoter capture Hi-C for, was to look at this concept of hubs, where a gene can interact with multiple enhancers, and those genes can also interact perhaps with other genes to regulate a transcriptional program at a more wide scale and coordinated manner. So here I have called hubs using the promoter capture data, and this is fairly straightforward, simple, but I'll so basically I broke down these hubs and looked at the gene connectivity. So how many for each promoter, how many regulatory elements are they interacting with? And also looked at the RNA expression. So this is just for one of the genotypes shown here for brevity. But essentially, as you increase in connectivity, you have an increase in gene expression. And also this, on the right side in blue, here is a similar trend, but in this case, as you increase with connectivity, you also have stronger interactions as well. The loop strengths are increasing, or they're supported by more reads being called. So these aren't not as transient as you would expect. They are considered strong and real. So here I'm just going to show a quick example, just visualize, because I think that also helps a lot. So here at the top, these loops are the promoter capture high-C data. Here, these are tag domains, and these are the C-scores signifying the compartments, where red is showing an open chromatin and blue is showing a compacted chromatin. And here at the bottom, and these things are the comparisons of just basically the delta for these compartments. So where red is up here, these are showing that the compartments between these comparisons are becoming more decompacted. And then here, there's just some ATAC-seq data to display like where there might be an increase in those interactions. And basically, like just from like a brief overview here, you can see that with these TAD domains, before in wild type and het, which are fairly similar, they are for the most part confined to their TAD domain. But once you get to homo, when there's more of the decompaction of the chromatin, you can see that there's actually an increase in these interactivity between TADs. So let's see, and just to look at, this is just zooming in on the SPRI2 locus, which is a stem cell gene. Here you can see this is SPRI2. There's an increase in accessibility at the promoter. And in wild type, there's actually no interaction. Het, there's like one interaction. It could be noise, it could be. But here, once you get to homo, so H1C and E are both homozygously depleted, you see that there's a strong amount of connections that are gained. And additionally, we do not have the K27 acetyl analytes yet, but to confirm that these are enhancers, but you do see that there's an accessibility increase as well for some of these anchors at the other end for these genes. So just to quickly summarize, basically H1 deficiency is going to increase these transcriptional programs by promoting several classes of genes, particularly those that are increasing plasticity through stem cell like neuronal genes and fitness, while also decreasing the genes that are in charge of organizing the chromatin and other genes that typically would be a safe block for more aggressive cancers. And then, so with the high C right now, we've gained that these compartments are opening, and it's a genome-wide type of shift. And here, opening particularly in regions where genes that are not off, but they're more of the mental type genes are located at. And I didn't show the TAD analysis here today, but there's also changes in TAD interactivity as well. And then for the promoter capture data, that's still a work in progress with further investigating hubs. But it's showing that the interactions with these promoters are becoming more connected elements that they previously were not connected to before the H1 depletion. Yes, and then this is just the acknowledgement slide. I'd like to thank my lab. There's a lot of people, so I just listed the two that like really helped me from both labs, and then some of the collaborators here, which have helped me with really brainstorming different ideas and such. So thanks.
Thanks for your patience. So our next speaker is Jack Satterlee.
And I can just use the controls on that. All right, so hello everyone, thanks for coming to the talk today. So my name is Jack Satterlee. I'm a postdoc here at Cold Spring Harbor Lab in Zach Lippman's laboratory. And today I'm going to be talking a bit about a project in which we're assembling chromosome scale Solanum genomes. So this is a genus that includes species like tomato to gain insight into the genetic basis of trait evolution, with a specific eye to the effects of large-scale structural variation and gene copy number variation, and how that's impacting traits in this genus. Oh, click. Okay, there we go. Okay, so before I jump into the work, I just wanted to say that this is a big collaborative project between many different people, both here at Cold Spring Harbor Lab, as well as our collaborators at Johns Hopkins University, Mike Schatz and Katie Jennett, leading the assembly of some of these very heterozygous and difficult to work with wild species genomes, and also collaborator Matthias Benoit in France, who's a former postdoc in the lab and really got this project started. And then also our collaborators at the University of Toronto here at CSHL, Jesse Gillison, Mike Castellacqua, his grad student. So why are we interested in generating these chromosome scale genome assemblies for the genus Solanum? So I think in most fields in genetics, there's been this really big new appreciation for the effects of structural variants and their abundance when you're looking across different genomes. And in tomato, that's no exception. So previously the lab did a large project to resequence genomes of both cultivated tomatoes, so the large fruited S. lycopersicum you can see at the top right, as well as the wild ancestor of tomato, S. campanellifolium. And what you're seeing here is that as we compare these genomes, these long read resequenced genomes, as we go from cultivated tomato to tomato wild relatives, there's this huge increase in structural variation that we previously were able to identify with short read sequencing technologies. And in particular, as you move farther out, you see greater and greater amounts of gene duplication. And we're also learning through these pan-genome analyses, for example, another study in tomato, that using these structural variants in genomic prediction helps fill in some of that missing heritability that's long plagued GWAS studies. And so we're particularly interested in these cases of gene duplication, so some type of structural variation. And because in many cases, this is part of this sort of hidden or cryptic genetic variation that we want to better understand how selection is acting on, how it's contributing to trait variation. So this is one example of this type of gene copy number variation and its importance in the context of domestication of tomatoes. So an important locus that helped contribute to the increases in fruit size we have in cultivated tomato is this fruit weight 3.2 locus. And these authors back in 2013 mapped this locus to a gene called CLU, which is a gene that promotes increases in organ and tissue size in plants. And so these authors attributed the mutant or the fruit weight 3.2 variant to a SNP, a causal SNP in the upstream region of the CLU gene that they suspected was increasing the transcript abundance and expression of this gene, leading to greater fruit size. But it turns out that when we went in and we sequenced these genomes, we found that the CLU locus is actually a tandem duplication of this CLU gene. So we have an increase in the gene copy number at this locus. And using CRISPR-Cas9 genome editing, former members of the lab actually reduced the copy number of this gene and showed that you get this quantitative response in fruit size as you titrate the amount of CLU gene in the genome. And so why are we interested in doing this in Solanum? It's not just because we work on tomato and we want to know what's going on in other species, but because Solanum is a really special plant genus. So it's one of the largest plant genera. It's in the top 10 of recognized plant genera and has over 1,000 species in it. So there's this tremendous amount of diversity that the genetic basis of which remains largely underexplored. And so there's a wide range of both cultivated and semi-domesticated species in this genus. So we have common staple crops like potato and tomato, as well as lesser known species such as pepino or naranilla. And we're really interested in understanding not just how trait evolution has impacted the different phenotypes of these plants, but also how we can learn from understanding the genetic variation behind these traits, how we can potentially target these species for improvement or neo-domestication. So as I said, this is a large project to get really high quality chromosome scale assemblies for these genomes. So collaborating with Katie Jennett and Mike Schatz at Johns Hopkins University, we've been using PacBio long-read hi-fi sequencing to sequence these genomes using a combination of Hi-Fi ASM and HETREC to get assemblies for these highly heterozygous plant species. And then in relation to the Arima workshop today, we've been using Hi-C as part of this pipeline to actually scaffold these genomes. And so this is primarily work that Katie Jennick has been doing, which I'm sharing today, but we've really gotten some impressive N50 scores for these genomes. And so we're excited to start exploring these. We hope that they will be really high quality for the community to eventually use. So this is sort of a broad view of macrosynteny across the different chromosomes of the different Solanum genomes we've sequenced so far. At the top, you have tomato, and then as you go down this list, you see the various chromosomal arrangements that we're seeing coming up in these assemblies. So this is looking at gene space macrosynteny. And so an important part of validating these assemblies is actually seeing whether these structural variations that we're seeing, these large-scale rearrangements of the chromosomes, are in fact real. And we need a way to validate them. So one example that I thought I would focus in on is a chromosomal rearrangement from chromosome 11 and chromosome 4 in tomato. They appear to be sort of fused into chromosome 4 and 11 in pepino or Solanum mericatum. And so what Katie's done using Hi-C data we've generated with Arima Genomics is to actually look at the chromosomal contacts in these different chromosomes to validate that the regions that are being assigned to chromosome 4 and regions that are being assigned to chromosome 11 actually interact with one another or interact with themselves more than one another, suggesting that we have a true translocation here in the genome. And we're also using this Hi-C data to identify potential misassemblies in the genomes. So for example, in this case from Solanum crenophyllum, which is an Australian Solanum species that we've sequenced, you can see here a clear misassembly where these DNA sequences have very low interaction rates with their neighbors in the assembly, suggesting that this is in the wrong place. So like I said, we're really interested in using these genomes to better understand the effects of gene copy number variation and gene paralog evolution over the evolutionary timescales captured in the Solanum genus. And so we've also been looking at shorter evolutionary timescales within a species. So in this case, Solanum aethiopicum, or scarlet eggplant, which is a domesticated species from sub-Saharan Africa, and it's also cultivated in Brazil. And we've been using long-read Oxford nanopore sequencing to better understand what haplotypes associated with copy number variation might be associated with traits that are both ecologically relevant as well as relevant to domestication. So here what I'm showing are several resequenced genomes of Solanum aethiopicum. And in these top purple read pileup maps, you have plants that are expressing this pigmentation phenotype. So they're accumulating high levels of anthocyanin, whereas these plants at the bottom don't accumulate any of this anthocyanin pigmentation. And so what we have these gene copy number variants associated with key transcription factors that we know to be involved in anthocyanin biosynthesis appear to have undergone many rounds of duplication in some of these genomes, and that's associated with this pigmentation phenotype. And what's interesting about this is it might be these amplifications of these transcription factors in these genomes might actually be driving differences in phenotype and rescuing the loss of pigmentation that's actually occurred in those aethiopicum accessions when you compare them to a closely related domesticated species like Solanum melongena. So in Aethiopicum, there's been a SNP that's been identified upstream with one of the downstream biosynthesis regulators of anthocyanins, this DFR gene, which has been shown to inhibit the binding of these transcription factors to the promoter, whereas in melongena, the binding motif for these genes remains intact. And so potentially by increasing the levels of this transcription factor through these higher copy number levels in some of these accessions, you can actually restore expression of these downstream genes. And so we can also look more broadly at this locus across multiple genomes. And we can, in these synteny plots here, these yellow arrows are these MIB transcription factors involved in the anthocyanin biosynthesis pathway. And what you can see are contractions and also some expansions of these gene duplicates at this region, suggesting that this might be a hotspot for this type of genetic variation. And so we're interested to see more broadly how that might be affecting traits. So another trait we're really interested in is related more closely to domestication, and that's changes in fruit size. So the same species, Solanum aethiopicum, so talking about with regard to the pigmentation, originated from a wild relative called S. anguivi. And so you can see that over the course of domestication, there's been this massive increase in fruit size, which is a very common phenotype, part of the domestication syndromes in other species as well. And so tomato is no different. During domestication, we went from very small tomatoes found in the wild relative S. pimpinellifolium, these much larger tomatoes that are commonly grown for processing in modern agriculture. And so it's been known that an important regulator of this phenotypic change is this gene called clavata3, which is a small secreted peptide, which negatively regulates fruit size. And so when this gene is mutated, you get this massive increase in the size of the fruit. And what we're learning is that there have been these very interesting copy number changes and duplications of these clavata3 genes across our different genomes that we've been sequencing. And these different duplications might have effects on the predictability of, say, genome editing if you want to increase fruit size. So whereas in tomato, you might have a very simple route to increasing the size of your tomato fruits. But in some other species, if you were, say, going to try to neo-domesticate Solanum phylum, you might have to counter the effects of having these duplicated copies, leading to some unpredictability in genome editing outcomes. And so in S. aethiopicum and S. melongena, these two domesticated species, you've actually lost function through pseudogenization of clavata3A, but have retained function of clavata3B. And so on shorter evolutionary timescales, we're also using our resequenced genomes to investigate structural variation at this locus. Okay, missed it. And so we're finding really interesting differences in this duplication locus, even over the short evolutionary timescales we're studying in the context of Solanum aethiopicum. And right now we're working on mapping some of these traits using QTL-seq. So the long-term goals of this project are to better understand over the evolutionary timescales in a single genus how duplicated genes have diverged in function or not, or have undergone pseudogenization. What are the factors that are driving these changes in gene duplicate function? And then also another goal is to assess which regions of the genome might be predisposed to yielding these types of copy number variants, and what signatures might be used to predict potential future sites of copy number variants and genome instability. So with that, I would be happy to take any questions. Yes.
This is maybe a really stupid question, but can you extrapolate any of this to mammalian genomes and copy number variations that are found in?
Yeah. I think so. I was just reading today actually a little bit about, for example, the Hox gene clusters, and I think that is also a really interesting system to think about. Duplicate paralog retention versus loss in different contexts. And I think what we'll ultimately maybe find in a lot of these cases is that, when we have a duplication, we'll probably see similar patterns of subfunctionalization based on loss of certain enhancer elements surrounding the gene, or sort of these compensatory shifts in expression that reflect that kind of divergence. The interesting thing is that we're looking over very short timescales, like in a single genus. So I think we can really get a sense of what are the early first steps when gene duplication happens. So yeah, I think we'll see a lot of similarities.
Are there ways for you to actually, somewhat randomly, but also partially intentionally, create duplications?
Yeah.
Affects things?
Yeah, so I mean, I've been interested. I mean, the coolest thing to do would be to do some sort of artificial evolution experiment. Fortunately, we're working with plants with long generation times, but I think it might be interesting to see how taking genes from species that have, experienced a duplication, and maybe that's associated with some sort of phenotype, taking one of the duplicates from that genome, bringing it over into one that hasn't experienced that duplication, and seeing how that might affect phenotypes or the function of its paralog in that genome or ortholog in that genome. So yeah, I think those cross-species experiments could be a way to